Thanks, everybody. Thanks for coming to the talk this afternoon. Right, I'm speaking on AI for drug design. There's a version of this talk I give which I call Hype and Hope in AI and Deep Learning. And the sort of further claim, like on the more cynical days, it actually looks like, deep learning, surely 60 million epicycles can't be wrong, if you guys remember your pre-Copernican models of the solar system. But concretely what we do is we use AI to help people discover new medicine. And so I want to actually begin by motivating it just with a couple questions that we answer for our customers. So here are like real examples of questions that we've been called upon to answer, to contribute to. So this first one is, we were talking to a pharma company, and they had a molecule they were super excited about. They had a piece of biology that they thought was really exciting, and it hit scaffold-based toxicity. And so all of a sudden they went from being 18 months ahead of all of their competition to actually being 18 months behind. And at that point, you sort of look around and you say, okay, we've got medicinal chemists and they've got medicinal chemists. We've got biologists and they've got biologists. We have high-throughput screening robots, they've got high-throughput screening robots. It can be pretty tough to see how you're going to leapfrog and catch up in that situation. So people are looking for new ways and exciting ways and some answer which gives you an edge beyond what you can do otherwise. And so hopefully what I hope to convince you through this talk is that AI and specifically machine learning approaches and specifically deep neural network approaches to the drug design problem are ways of doing that much more efficiently than has been possible in the past. That's the official goal. Like the unofficial goal is you should all come join Atomwise, but like that's just between you and me. Don't talk about it. Second example is sort of related, and this is one where targets go in and out, like disease targets go in and out of fashion. And a lot of what that's governed by is what people believe is possible, like what the preconception is. And so if there's some breakthrough or some insight that gives a new handle or a new binding site, a new way to go after these things, then all of a sudden there's this land rush to start working on it. And so every new technology may swap things on the border. What was previously impossible is suddenly possible. And so our customers talk about, are there undruggable targets that we could make druggable with these new technologies, and thereby open up whole new avenues of therapeutic opportunity. And that's sort of similar to the other one. Can we be first? And then relatedly, just how do we do more faster? How do we get after? So these are a couple of the concrete conversations I've been having out there with people who are trying to discover new medicines. And our answer to each of these is AI, which I think is the answer of this whole lecture series. And so then this brings me to this hype versus hope thing, because there is a lot of hype out there. I think a previous speaker said that AI is the new electricity, which is a nice, fun claim. CEO of Google says that AI is more profound than fire, and that is really quite a bold claim, I think. I think it's interesting when you see things like AI is the new electricity to think back to when electricity was the new electricity, right? Because there might be lessons learned there. And so electricity certainly has transformed our lives. It was absolutely fundamental. But here I want to turn to factories. And there were factories before there was electricity. Here's a depiction of steam factory. And you can see that here just like the central organizing principle of this factory was this line shaft, this thing overhead, right? Which is just an engineering solution. It's how you move power from the boiler room down to the basement to each of the machines. And the whole organizing principle of the factory was around this engineering solution, this engineering constraint. It actually took about 50 years after the development of electricity to first decouple these machines from the line shaft. And then you had this transformation where you reorganized the factory around the workflow, because you could move the machines. And that actually took 50 years after electricity, right, to get Henry Ford's assembly line. Because that requires a change in mentality of how people think about work rather than technology, right? And people's mentality always changes much slower than technology changes. And so like one central theme of AI, I think, is that achieving these true productivity gains, what's possible with AI, is actually going to require redesign. And that's the hard part of it, rather than developing new technology. So for the people in this room who are interested in working at these intersections of AI and healthcare and how those intersect and what needs to happen, I think what you're going to find as you try to make change is we're actually going to have to change the way we do things. In drug discovery, boy, we could use some redesign, right? Because I think modern lab work would look depressingly familiar to people, to scientists from a century ago, right? And in fact, I would say, I mean, the Atomwise claim is, this is a little bit weird, right? That fundamentally pharma is the last industry where you build every single prototype before you build a prototype, before you test the prototype. That's unusual, right? And that's the thing that we're trying to solve with AI at Atomwise. So what do I mean by that? If you are Boeing or you're Lockheed Martin, you will simulate 1,000 wings before you ever build one wing and put it in the wind tunnel, right? And it's only after the computer says, wing number 212, it will fly, it will be fuel efficient, it will be quiet, maybe a dozen other factors. Only after the computer gives you those answers do you bother building the prototype. And then you still run the test. You take it to the wind tunnel, you still do the test flight before I ever get on it, I hope. But you do that after a whole lot of testing has been done with the prototype, rather than before, rather than up front. And similarly, you know, we live in California, we have pretty good confidence that this building will stand up in an earthquake. But that's an expensive experiment to run, right? We don't run the experiment first, right? We run the simulations first to give us that confidence. And so you could go to Boeing and you could say, I've got this brilliant idea. What we're going to do is we're going to throw 1,000 different winds off this cliff. And then we'll just experimentally know what will fly and what won't fly. We can do it in this hypothesis-free way, and we won't be constrained by our prior preconceptions. And that would work. They just won't let you do it. It's like just way too expensive. And so as you go through, it doesn't matter if we're talking about Boeing, Lockheed Martin. It doesn't matter if we're talking about, you know, building a dam or building a new power system. If we're designing the Tesla, if we're doing, you know, structural engineering on these buildings, before Intel drops $1 billion fabbing a new chip, they have a pretty good guess that it's going to work. And so pharma is kind of the last one where you build every single prototype and you actually physically test it. That I've been able to think of. Maybe keep me honest. I mean, like, tell me if I'm missing big industries where that's true. And so I think that means that there's a real opportunity there if you can get predictive performance high enough. Make sense? Great. Okay. So that's what we're working on. And so here, I was told that, you know, there's some people in the room with a lot of life science background, there's some people in the room with a lot of computer science background, there's some people in the room with both, and so, you know, again, keep me honest about my claims here, but here I wanted to just give like a 30-second crash course into biology and what the concrete problem that we're trying to solve is. So you can think of, for people with absolutely no bio background, you can think of the proteins in your body as machines on an assembly line, where each machine takes in a very specific input, transforms it a specific way, hands it off to the next machine, and on down the line. And so out of, you know, the food you eat, your body breaks that down and then builds more of it. So one of the ways that disease happens is when those machines break or when they go haywire. So if the machine which governs cell growth and division switches on and it never switches off, then that cell keeps growing and dividing, that's a tumor, that's cancer. Okay. And so if you saw a machine on an assembly line going haywire, you might throw in a monkey wrench into that machine. So the machine is busy chomping on the monkey wrench instead of doing whatever it normally does. And that way you shut down the machine and you fix the problem. And so that's the way a lot of drugs work. And here I'm, you know, giving this analogy, but if you look up the Philadelphia chromosome, that was the target of the first rationally designed drug, Gleevec, imatinib. You'll actually see that it is really just that. It is a protein which governs cell growth and division that gets switched on, never switches off. Here I'm actually showing in purple a protein, and in yellow I'm showing one of the oldest chemotherapies, methotrexate, back in the 1940s, 50s. And you can see that it just like, there's this groove in the protein, it physically occupies the site, and it prevents whatever the normal input to that molecular machine to enter, like that monkey wrench. So, for developing a next generation of chemotherapies, you want to know which of these bumps and ridges and interactions is idiosyncratic and which ones are absolutely critical. Because you want that monkey wrench to bind in very, very strongly to that machine and shut down, so you have to use very little of the drug. You also want to make sure that you shut down this one protein, not 100 other proteins that cause all manner of side effects. And so it's super critical and super important to know which ones here are the important ones, which ones are not. Because I think the idea that very small changes in shape have huge differences in how well things fit together is actually quite intuitive for all of us. And so in addition to shape, you need charge and complementarity and hydrophobicity and hydrogen bonding and so forth. And so I'll get to that in a second. But so that's concretely the goal. And so what we do is we help judge which ones are going to fit well, which ones are going to bind, which ones don't. And you want the molecule to bind the intended disease protein. You want to bounce off proteins in your liver, your kidneys, your heart, your brain, things you want to keep functioning the normal way. And so that's what I'm saying up there, that by binding, it gives you a handle on both efficacy and safety. Okay, off-target tox. Okay, so that's a challenging problem. And here I want to give you an AI parenthetical, because I think there's some business students in the room who will be called upon to evaluate AI business plans. Like probably everybody got a summer internship off on Sand Hill Road, right? So you're going to have to look at 600 of these things and decide which one's fun. So here's my AI parenthetical. You can look at, and I think I actually may have sold this one from Andrew Ng. So credit where credit's due. I think in one of his talks he talked about this, which is it's much easier these days for AI to be cheaper or faster than humans. It's still very, very challenging for AI to be better than humans. Right? It's possible. We can achieve, and sometimes you see this probably in some of the other lectures, you'll see diagnosing retinopathies, for example. Diabetic retinopathies can actually transcend human level ability, human experts. But that's really quite challenging. Dota 2, maybe there are people in the room who follow that work, right? Like we can beat humans, but mostly what AI these days still can achieve is just being faster or cheaper. And so one way of sanity checking a business plan is if unlimited humans don't say the business plan, then the AI won't either. But it's absolutely the case where machinery can do way better than the previous human approach to it, still has incredible value, right? Because you can achieve previously unachievable scale, and that unlocks all manner of value. And so how does that look for us for drug discovery? So here I'm showing the number of molecules that you could purchase out of a catalog. Okay, so back about a decade ago, there were maybe 1 million molecules if you put all the vendor libraries together, which we in this room could purchase. And at the time, Big Pharma had between 3 and 5 million molecules in their internal corporate collections. So that meant that we were at a disadvantage, you and I, right? Which is that Big Pharma had a better chance of finding something that interacted with that disease protein. And having found that thing, by having an army of medicinal chemists, you could iterate your way to a drug faster. And so scale mattered, both scale of robots and warehouses full of freezers and your corporate collection and hardworking, judicious, insightful medicinal chemists, right? It was having the army mattered. Recently, that switched. And so the catalog, the synthesized on-demand universe, and I can get into why this has happened, and this is a log scale, by the way, but the incredible growth in chemical manufacturing that we've seen over the last 2 decades in places like India, in places like China, in places like Belarus and Ukraine has shown just incredible amounts. And so here I'm showing actually just from a single vendor, enemy in the Ukraine, they offer synthesis on demand 3.8 billion molecules. So, 1,000 times what Big Pharma topped out at. And so all of a sudden, this means that the real bottleneck, the real constraint, this chart should be absolutely terrifying to Big Pharma's Pfizer, Merck, GlaxoSmithKlines of the world, because what it used to be the core strategic competitive advantage of having these armies of medicinal chemists, all of a sudden finding molecules and constructing molecules isn't the limiting factor anymore, but it's which molecule is the right molecule. Right? As manufacturing gets easier, it shifts from finding to matching, right? From making to matching, from from synthesis to searching, right? Like, if you go on YouTube, there is way more content than we could ever watch in our lifetimes. And so the question is not, oh, there's nothing on TV, right? The question is like, which thing should you watch? What are you going to find funny? And that becomes a machine learning problem and not fundamentally a physical manufacturing problem. So I think this also highlights both the challenge of computational techniques of the past, right? If you were here, maybe you didn't have the right molecule in the set to find. But it also highlights the challenge of computational techniques of the present. Suppose you build an incredibly accurate AI system. It's 99% accurate. Okay, sounds pretty good. Suppose we go even further, that it has a 0 false negative rate. It will find the right answer. Okay, it will return it. But it has a 1% false positive rate. Okay? What that means is, out of the sea of 3.8 billion, you'll find the right molecule. It will be in there. But it will be lost in a sea of tens of millions of molecules, far larger than the entire corpus collection. You'll never actually, for practical reasons, find it. And so this means that your false positive rate has to be, like your accuracy rate has to far exceed 99%. It has to be 99.999. Just pick your number of nines that you like. Like, the more accuracy you can get, the growth in chemical universe will exceed them. So you've got to have exceptional accuracy. And so the whole question becomes, how can you deliver that? So let me speak to how I can. So we're not the first ones to say computers for chemistry are a good idea. So there have actually been generations that sort of came through. And the first set of people to come through were physicists. And they said, look, I can simulate physics, right? Like, what's the problem? You know, I can compute van der Waals dispersion, I can compute Coulomb charges, right? Like, the cows are spherical, right? Like, this should be a pretty simple problem. And it turns out that if you do full quantum mechanical simulation, and Professor here got the 2013 Nobel Prize in Chemistry for doing exactly this back in the 70s, if you do full quantum mechanical simulation, you get the right answer. I find that comforting. I'm always comforted when, like, physics actually works. And so that's the good news. The bad news is the techniques scale factorially in the number of electrons in your system, and so it gives you the right answer if what you're interested in is lithium hydride. And it just doesn't scale to the kinds of things we're interested in, which is interactions with biology, the size, the scope of biological questions. And so people don't do physics. At best, they do things which are inspired by physics. They do approximations of physics, physics-esque or physics-like. And so the next generation came in and said, I don't just know physics, I also know some chemistry, and so I'm going to actually teach the computer chemistry. And so the second set of people who came through said chemistry. So I'm going to teach the computer how to detect hydrogen bonding because I learned hydrogen bonding in my organic chemistry class, and so I'm going to, I'm going to teach it how to recognize a donor, I'm going to recognize an acceptor. I believe that the distance matters between those two. And then you say to yourself, okay, it's not just the distance, and it's not a simple function, there's a domain of repulsion, a domain of attraction, but you can write that down. But then you think to yourself, okay, the angle matters, both in and out of the plane, right? Like, if that's a strong bond. And now you start getting depressed, right? Because the angle is not actually a parameter itself, it's actually a function. The preferred geometry changes if this thing is in ether, if it's a carboxylic acid. And then you start getting really depressed and want to go into finance instead, because it's not just the distance and what's interacting and how they're interacting, but the environment in which you find yourself also matters, right? Like, are there stabilizing or destabilizing charges? Are there resonance effects? Is the hydrogen bond part of the hydrogen bond network, or is it solitary? It turns out very quickly we have to admit to each other that there's not a hydrogen bond you're teaching the computer, it's that there's 100 different hydrogen bonds depending on context. Okay. Turns out that is actually exactly the same story that happened in computer vision. Okay, so a classic problem in computer vision is facial recognition. And so there, the low-level features aren't hydrogen bonds, but they're side-of-face, edge detectors, right? So like, is this pixel my ear, or is it the wall behind me? Right, so we're going to take a photo of Neil, we're going to take a photo of Abe, and we're going to ask the computer to say who's who. And so it really matters if we can, correctly classify the edges of the face. Okay. And so exactly the same scenario happened, right? Where that pixel looks different under these lights than outside in the sun. It looks different if a shadow falls across my face. If I'm against a light background or a dark background, it's going to look different again. And so again, we have to admit to ourselves that there's not a an edge detector, a side-of-face detector, is that there's hundreds of different ones based on context. So what people used to do for decades of computer vision is you would sit there, you would like manually whittle edge detector, you would do feature engineering, right? And the art of machine learning was actually feature engineering. And that basically stopped in 2012. And in 2012, deep neural networks were invented, discovered, where what that is a means of statistically deriving those low-level feature detectors rather than teaching the computer explicitly. And when you do statistical derivation, then instead of the last 3 examples that you looked at, which is frankly all we can like remember, that's like all we can fit in our head, instead of like looking at the last 3 examples, the last 3 photos, for example, the last 3 protein ligand structures, you statistically optimize for the last 3 million examples in your training data. And not only is that way more accurate, importantly, critically, it's way more robust, right? It used to be the case that, at least in computational, in computer vision, what we saw is in the lab, there were technologies which, you could do facial recognition in the lab, but you couldn't unlock your iPhone with it, right? We had robots in the lab, but we didn't have self-driving cars on the road, because the robustness had to be under these carefully controlled conditions. And so that's been the transformation, is that robustness. Drug design, what we had in computational chemistry, and I think it's fair to say, is you had this endless series of overfit energy functions to the particular protein that was being studied. And you could get it to be predictive, but it fell down when you tried to do it for real drug discovery. It was a perfect model if what you wanted to do was graduate. If you were like a grad student and you needed to graduate and you needed papers for your PhD, you could like endlessly overfit energy functions and publish on it, right? Like, and so the system was designed to solve the problem that like the practitioners had, but not the problem which maybe we in this room care about, which is going out and really doing discovery for real. Okay, so how do you go from edge detection to molecular binding? Images for a computer are a 2D grid of pixels, and every pixel has red, green, and blue color channels. Proteins are 3-dimensional, and so we set up a 3D grid of volumetric pixels. And every voxel, instead of having red, green, and blue, we have oxygen, sulfur, nitrogen, carbon, sp2 carbon, sp3 carbon for the chemists in the room, channels, color channels. And as soon as you do that in coding, then everything which Andrew and Feifei and Jan and Yoshio and Jeff are inventing for images, we get to translate into 3 dimensions instead of red, green, and blue, carbon, oxygen, sulfur, nitrogen, and we get to run in 3D for drug discovery, for predicting small molecule protein binding. And so down here, twirling, instead of edge detectors and higher order features like eyes, nose, and mouths, what we get is sensible biochemical features. And so this is some data that we presented at the American Chemical Society, and here we picked up a particular neuron 614, and said, hey, neuron 614, what in the input maximally activates? And you can see that we've drawn this box around the voxel, which was maximally activating that neuron. So you'll see over and over again, as these things flip through, that it's firing on the sulfonyl group. So it has learned this particular configuration of the yellow and red sticks, which throughout the training correlates either with binding or with non-binding. It's actually a structural feature which shows up in quite a number of antibiotics. But it is so reasonable that we actually have a name for it, right? We call that the sulfonyl group. And remember, all that we were giving the neural network was X, Y, Z, and atom types, right? It was just X, Y, Z coordinates and sulfur, oxygen, nitrogen, carbon, whatever. And it has decided that this particular constellation of points in space makes sense. Other examples that we looked at were things that your friendly neighborhood medicinal chemist will tell you are reasonable, things like aromaticity, single bonds, so rotatable bonds, metal ions. And actually we showed that there are neurons deeper in the network which fire when the metal ion and the sulfonyl group are near each other. And so if I can amorphize this for a second, the concept of metal coordination seems to be dropping out purely statistically and detectable in these in the network, just in the activation of the neurons of the network. This is some More recent work, and this is sort of now translating, that was the theory, that was the architecture of the neural network and so forth. And so here it's prospectively, this is work that we did, presented at the last American Chemical Society meeting, so I can tell you that this is work with Genentech just up the road here. And so here this is, they built tens of thousands of real medicinal chemistry compounds, doing real discovery on projects running up for a long time across a diversity of protein classes. They gave us some of the data, and then we wanted to predict activity. So let me just, in the interest of time, get into it. This is some data that, internal benchmark that we built, but you can see that useful as a drug is this 1 nanomolar, so it's this vertical line. And so this is a histogram on like real experimental potencies. So everything to the left of here was too weak to be a drug. And so you can see that the vast majority of experiments in the training sets are never going to be drugs, so they just don't have the potency. And so then here on the y-axis, I'm showing the predictions. And you can see just by eye that there's this really nice correlation. More than that, if you focus only on the ones which Atomet thinks are going to be highest potency, then actually you avoid a lot of that work, a lot of that wasted work, which otherwise, would be the majority of these experiments. This chart is aggregated across those different proteins, so here I want to break it out. Let me explain what I'm going to be showing you. So on the x-axis, these are 9 different targets of medicinal chemistry interest, so 9 drug discovery targets. And what I'm showing on the y-axis is mean absolute error, sometimes called mean unsigned error. And so the way this goes is, if you measured in your test tube that a particular protein, a particular molecule bound at 1 nanomolar potency, and I said 3 nanomolar potency, that would be a 3x error. Okay. And so that, by the way, 2 to 3x is if they ran their assay twice, their experimental error, they would see sort of 2 to 3x error between runs of the assay. But that's if they ran it, right? So if the same person on the same equipment with the same assay, the same laboratory runs it, you get sort of 2 or 3x error bars. If you work with an external group, they want to see 5x error bars or better. Okay, so this is working with an external group. And then 10x, so like, I measure, or you measure 1 nanomolar and somebody said predicts 10 nanomolar, so 10x difference. That's kind of been the boundary that's been very, very challenging historically for computational methods to transcend. So that's 1.4 kcal per mole for chemistry. So here's how we do. And in particular, for these 3, you can see, so C, Charlie, F, Foxtrot, and H, Hotel, you can actually see that our error bars are indistinguishable from working with an outside group, right? So real physical experiment. And for 8 of the 10, A through H, we actually transcend this computational bound that's been very challenging for computational techniques in the past. But at the time that we were doing this, we were able to deliver 40 million molecules a day. And these days we can do about 100 million molecules a day. And so that's much vaster swaths of being able to interrogate computational space much broader than has been available in the past. All right. I want to sort of talk about how difficult it was to do this. These are impressive results. And what I want to do is sort of talk about measuring AI techniques. And so again, here I have an AI parenthetical, which is like, this is Mickey Mouse, his interaction with AI. Okay, so do you guys know the Sorcerer's Apprentice story? Possibly everybody here plays the piece in, anyway, whatever. Okay, so this is the one where like, Mickey is the eponymous Sorcerer's Apprentice, and the sorcerer leaves and he doesn't want to clean the place, and so he conjures up and animates the broomstick, the AI, and says, hey, what I want is water, go get me water, okay? And so the broomstick gets water, but like gets way too much water, right? Just maximizes for the error function that was set before it, and like solves the problem, but like doesn't obey any of the constraints of what Mickey meant instead of what he said, okay? So you guys have probably seen the OpenAI thing where like they had a speedboat racing game, and or the OpenAI one where they discovered a bug in Qbert that like if you got Qbert to jump off the board, you got infinite points. And so this game didn't learn how to play Qbert. What it learned was how to exploit bugs in the underlying ROM, like in the underlying simulator. Okay, so this is Mickey Mouse's version of that. Okay. This is the last paper that we published. It's titled "Most Ligand-Based Classification Benchmarks Reward Memorization Rather than Generalization." Okay, that's the title. Catchy, I know. But this is the problem. So the punchline that I'm going to show briefly is we looked at a bunch of benchmarks. In my opinion, every single one of those benchmarks failed to separate whether AI systems were overfitting to the test versus being accurate on the test. Every single one. And so, if you believe my results, like I do, then this means that every result in computational chemistry that we've seen for these things is likely to be incorrect. Because there's way more ways to get it wrong and overfit than to be accurate. Okay? So again, if you're a grad student and you want like a bunch of citations, then we provide code for unbiasing benchmarks. And so somebody should just like go and bias the benchmarks and like redo every assessment that's been done in this field because it's probably been wrong. Let me get into how. Okay. Here's, I've got some positives, I've got some negatives, I've built a classifier, it does really well. Thanks, whoever did that. Right? And so I've built this classifier, and it's great. And so now I go and I want to see whether it's actually useful, and so I go test it on my test data, which is here in blue. Okay? Red, excuse me. And so if you are red-green colorblind, you cannot tell the difference between these. But neither can anyone else. That's the point, right? Like here, these are basically identical. You've not actually chosen an independent test set. And so you're gonna think you're doing fantastic, but it's actually just overfit your system. And so what you really need is you need like really actually novel data that generalizes in the way that you care about. And in the novel data, you might say, oh, yeah, okay, the simpler predictive model is better. The simpler predictive model, the more complicated one was overfit. What I thought I was doing was overfit. And so what you need is this really novel data rather than highly redundant data. And it turns out, and this is a chart from a paper out of Vijay Pandey's lab. And so you can see that like here's what they're doing is the similarity with a couple different splits, where one is identity. And you can see in either the green split or the blue split, there's actually like a lot of identity, a sort of surprising amount. And random, two random molecules, random molecule in this metric space is 0.15. So they did was way more similar to each other than random molecules. And so what we did, and not getting deeply into this paper, but what we did was we defined a very simple measure of what we call bias, and then we show here's a bunch of targets ranked by bias, here's random forest, SVM, logistic regression, k-nearest neighbor, here's their accuracy on this ordering. So you can just see by eye that as bias goes up, performance goes up, and here we actually do the correlations. And so what we're arguing, and this holds with every unbiasing measure, every benchmark, every machine learning algorithm we look at, everything we looked at has this trend. And so we think that this simple bias that we compute actually is what is being measured instead of real predictive accuracy. So once again, most ligand-based classification benchmarks are more memorization rather than generalization. And so like there's tons of ways of getting this wrong. And so the only thing that you can do is do it for real, go discover some stuff. And so this is what we've got to actually show. I think as computational chemists or people who are interested in AI applications for healthcare, the burden is on us to actually go prove that it works for real when you try to do these things. And so here's just a couple examples. So we did a project on Ebola, and so here's a particular protein which exists in this open conformation and then folds in on itself as part of viral fusion entry and closes up. So the idea was, can we park something roughly there to block Ebola entry into cells? And so here you'll see like that central piece, we are testing different things. And so each of these poses is that's scored by the neural network. The final score is an ensemble score over these individual poses. as weighted by the Boltzmann distribution. Long story short, we found one molecule which had blocked Ebola infectivity, at least in the cell culture, by about 80%. It also correctly failed to inhibit a virus with a different entry mechanism, so that's some evidence that's right for the right reason, even though these are phenotypic screens. And the interesting thing is that like the compound had no previously known antiviral activity. And that's important because a lot of what these AI systems, especially some of like the literature-based ones, is they'll say, oh, I see you're working on Ebola. That's a flavivirus. Have you considered dengue? It's also a flavivirus. All of these treatments for, all these drugs for dengue. And you show it to a virologist and they're like, yeah, thanks. Like, that's the first thing I tried. And so that's actually an important subtle point, which is that you don't actually get rewarded for accuracy. What you get rewarded for is accurate surprise. That's a much higher bar for AI systems than just accuracy. If you bore them, they don't care. This is a multiple sclerosis project. It was a novel protein interaction in the central nervous system. So it was two proteins. What our collaborators had shown before we met them is they block these two proteins from coming together that were on the surface of the neuron, that good things happen for their MS mice. And the way they had done that was they had chopped up each of the interacting pairs into peptides, and so you dose the peptide, it blocks the interaction from coming together. They tried to commercialize this, and my understanding is the pharma partners that they were trying to commercialize this to said, look, peptides don't cross the blood-brain barrier. Your brain is covered in armor called the blood-brain barrier, and they said that they don't cross the blood-brain barrier. So it's fine for you to directly inject the brain of your mice. We're going to have a patient compliance problem. Right? So we're not interested in that as treatment. So this is actually a really interesting case, which is not that uncommon, which is the biology was super well worked out, but you didn't have any chemistry to enable a medicine, right? Like that's the gap between research and helping a single human life, right? And so what we did was we screened basically everything you could buy at the time. At the time it was only 8.2 million were sitting on the shelf, much smaller than they are today. And we filtered out aggregators and autofluorescers and other things that were going to mess up the assay. And just skipping ahead, let me, in the interest of time, skip to, we found 9 binders in the top 50, so quite a good hit rate. After some follow-up, one was chosen to be dosed in mice. And I am not a biologist, but I have a happy-looking mouse and a very sad-looking mouse. Even I can tell that, with dragging hind limbs and limp tail, sort of the classic symptoms of multiple sclerosis in the EA model, and just very nice statistical separation between the treated and the untreated arms. So this is the kind of result I think that, usually when I'm presenting, medicinal chemists are like on their phones or computers. So we've got some medicinal chemists in the room. But like at this slide, they're like, oh, right? Like start over, tell people who you are and what you do. It's like, this is the slide where they start paying attention. We're actually making that available to academics around the world for free. So imagine, if you will, you're a professor at Stanford, and you think protein X, whatever it happens to be, will cure Alzheimer's or cancer or tuberculosis, whichever one it is. So we have like a very simple grant application on the webpage where you say what you want to work on. It's designed to be copy and pasted out of your other grant application, so it's supposed to be super easy. Just lets us know that like there is a protein structure and some other things we need to do our work. And we will go screen every molecule that's sitting on a shelf. We will rank them. We'll do our analysis. We'll rank them. We'll buy them. We'll quality control them. We'll plate them, and we'll send you a 96-well plate for free. And so you can just like run your assay and then go publish. We don't put constraints on the publication. And so what we're trying to show is democratize and enable discovery and people just to prove to themselves that these AI approaches really work. It's the logical conclusion. So if you search for Atomwise aims, please tell all your friends. We have over 100 projects going through the pipeline now. We just closed our third round of applications and had over 200 applications there, and we'll be announcing another one in the spring semester. So It's something, it's a way that people are finding things. So with that, that was the end. I know I was running late on time, and so I am happy to take questions. Thanks for your attention. Anybody? Yes, sir.
I have a question, I guess. The unbiasing research you were talking about, and going back to the genetics study you guys did, are you saying that your unbiasing algorithm was able to get the results that it did? Or is it that those results have to then go and be re-looked at to make sure that there wasn't bias and using the assumptions afterwards?
Great question. Okay, so let me see if I can rephrase just to make sure people heard. Is it fair to say, so the question was the unbiasing algorithm responsible for finding the results? Or what was the second part?
Or is it the unbiasing thing you guys realized afterwards, like, you know what, this is a problem that we didn't get good results, but it could be due to the fact that we're overfitting our data and our algorithms, and so therefore we need to find a solution to that.
Yeah.
What was that application solution?
Okay, right. So, okay. What was the genesis? Was it that we started with the unbiasing, or was it that we discovered it through our efforts? Yeah. So the unbiasing algorithm doesn't generate the results, right? The unbiasing algorithm is how do you test the test? How do you decide that the test is predictive, right? Because this is a general problem for people who are doing AI work is you have to set up something internally, a proxy for your future performance. And you're building your system usually against running that proxy, and then you hope that that proxy is really capturing how it will work when you take it out into the real world, right? And so you've got to test the test. And really what we saw is there were people out there who were publishing papers, and it would say in the abstract, it's like, we build our quantum Fibonacci analysis, whatever the future technique. We have deeper neural networks. We have deepest neural networks, right? Like whatever it is. And we're achieving 99.996 accuracy, right? And you say that in the abstract. Okay. The second sentence of that abstract could go one of two ways. The first way is, I'm a genius. That's universe one. Universe two is, and because we know that the physical experiment does not have 99% accuracy, we know we've overfit to the data, and so we deeply apologize and we're working to fix that, right? Like, there are two ways that those papers can go. Everybody was publishing number one, right? Everyone was saying, I've achieved 99% accuracy, hire me. And then 6 months later, another group would say, well, we achieved 99.9% accuracy, give us the grants. And then people were beating it, even though everybody had way far exceeded the actual error bars in any physical experiments. And so the way we found it was, we had internal benchmarks and baselines, and we were just doing like k-nearest neighbor, which is like a super simple machine learning approach for the non-machine learning people in the room, just as a baseline, just as a sanity check. And k-nearest neighbor was getting really high accuracy. And so either you can look at that and say, wow, we do k-nearest neighbor amazingly, let's publish. Or you can say, okay, there's a real problem with this test. The test is a bad test, right? And so then we're trying to quantify why the test was a good test, why the test was a bad test. Because for us, it's super easy to, there's two notions, right? There's a notion of motion and there's a notion of progress, right? And it's really easy to be pedal to the metal in neutral, right? And if you don't set up your internal benchmarks correctly, then like your AI system is going nowhere and you don't even know it, right? And so that's actually where it came out, was that we saw performance which, look, maybe it's just that we're like Eastern European and deeply pessimistic and skeptical, right? Like and depressive people, right? Like, but we were like, this doesn't make any sense. It can't possibly be that good. Prove to us that the world is terrible and none of these results are ended. So, but that's also how you do good science. Does that answer the question? A little bit too much, maybe? Great. Anyone else? Yes, sir.
Thanks so much for the presentation. I had a question when you think about this, not necessarily from the drug discovery aspect, but implementation as far as clinical trials, etc.
Yeah.
Within the pharma industry, might have drugs that seem pretty effective, but then you spend hundreds of millions of dollars to run these large trials to see is there some safety signal that we don't know about, whether liver tox, heart, whatever it might be that where people cross their fingers there isn't. Is there a way to accelerate that aspect of it on the safety side as opposed to just the efficacy drug discovery?
Sure. Okay, so the question again for people who didn't necessarily hear it was, you're talking about preclinical, but there's this huge cost and time sink in clinical. Can you comment on machine learning applications clinical? Fair we say that? Okay. You're absolutely right. Clinical is expensive. In aggregate, let me say, there's a paper from, I want to say Eli Lilly, it's Paul et al. from 2010, and they actually, they compute costs for each of the steps. And according to their numbers, actually, lead optimization, which is the problem I was showing you with with the scatter plot that went like that, and the arrow and the bar chart. Those charts are applicable to a processing drug discovery called lead optimization. They actually compute that that's the most expensive part. Because you have attrition of projects as you go through the pipeline, and so you run a clinical trial for the one project that survived to that point, whereas lead optimization you're doing across all of them. So in 2010 dollars, they were $414 million for lead optimization, $314 million for phase 3 clinical trials, which we generally think of as expensive. But undoubtedly they're expensive, right? Like they're the number two. And so if we had predictive tools that would say this is going to be effective, right? This is going to be safe, this is going to actually work in humans, that would be fantastic. Today in drug discovery, we talk about things like animal models, right? Like that's a phrase that comes up. And let me say, I don't work in clinical trial design, so I may be underestimating the sophistication of it, and any progress there would be hugely valuable, and we don't do any work in clinical trial design. But my understanding of animal model is the model part of animal model is mouse dead, mouse alive, human alive, which is not much of a model the way I use the word model, right? Like machine learning model. And so there's a lot of other stuff we could measure about mice, which you would hope would be more predictive when it gets to humans. And I mean, that's an exaggeration, but like it's closer to that end than like deeply sophisticated machine learning models. Here's my problem with working in the clinical space, because clearly the need is great. There's a lot of super smart people who've worked very, very hard to design clinical trials as well as we can. I think the problem is the amount of data is very low, right? I think there have been, through last time I checked, through phase 2 or better, so phase 2, phase 3 approved, there have been 8,000 drugs that got that far, total. 8,000 is not a big data set for deep neural networks, right? Like it's not a big data set for modern machine learning techniques. And that's across all diseases, right? And so I think it's a data paucity, that's a data scarcity problem. And so I don't know if there's some data sharing, if you put together all of the pharma companies, it's still 8,000, right? And so within any one organization, and then as an outsider, right, you don't even have those internals. Anyway, I think the biggest problem there is how you get access to enough data to like build those models. But it would be hugely valuable if you could do it. That'd be great. Yes, sir.
Question brought up you guys' business model a little bit. So the way I understand is you guys create sort of a platform, get the data set. Are partners accessing the data set, or are they accessing kind of like as a platform, or is particularly like your consulting services? So how do you guys see, like, do you own like the molecule that you discovered? Do you guys get a piece of like whatever will come for that potentially? Or I guess how do you guys see you guys like, in terms of scalability and kind of business model?
Sure. So for the people who couldn't hear, is it a fair statement to say your question is how do you get paid?
Sure, yeah.
It's a good question. And I had a board meeting last week, believe me. People want to know. The way that we work with people is really as a partnership model. So there's lots of people out there who are excellent biologists, right? Who have, you're really, like if you're doing drug discovery, you're solving one of three problems, right? Either you're solving problems in finance, how do you pay for all this stuff, right? And that's a hard problem. So I don't know that anyone has, deep insight into how to do it. The two technical problems are like, you're solving problems in biology, you're solving problems in chemistry, right? And so the problems in biology are what is the protein which is going to map to unmet medical need, right? Like which pathway maps to unmet medical need? Which protein in that pathway is the linchpin, is the really important one, and which ones are compensatory, right? Having just found that one, which assays have like nice clean error bars that we can measure anything about. Having found that, like which animal model is going to generalize to humans, like we talked about. Like, so all these questions about how you actually define what to go after. Because if you get that wrong, it doesn't matter what else you do, it won't be effective. But then having found that protein, having built the assay, having chosen the animal model, having done everything, you don't have a drug, which you have no drugs and you haven't helped any patients. And so then you need the molecule which modulates that protein, right? And safely, doesn't cause side effects, and is new and hitherto undescribed in the history of patents, right? Like, so that you can get paid to, right? And so the way we found that works best is for us to handle the chemistry and to partner with biologists who handle the biology. And so the AI system informs our medicinal chemists, who up to and include go and project manage the external contract research organizations to get physical compounds synthesized, right? To invent new chemistry, get compounds synthesized, and then we send it to the biologists, right? Quality control and so forth. Send it to the biologists so they can focus on testing. And so it ends up being, co-ownership with a number of different styles, whether it's with 50-50 joint ventures, whether it's milestones, whether it's back-ended stack royalties. The how you become partners changes. Different people have different preferences, but that working together is what's worked best for us.
Looking back to your example of two companies with the same resources, but one that have edge using AI, how would a AI startup in focus learning on edge because of pharma or using AI?
How would a startup using AI have an edge against a big pharma using AI? Well, it depends what they're using it for, right? Can I be a mathematician for a second? I believe there is the answer to your question is a lemma, right? Which is, or like a corollary to a much stronger claim, like a much stronger theorem, which is like, today without using AI, without any conditioning on what tools the systems use, there are big pharma, there are small pharma, and big pharma goes and purchases all manner of small pharma, right? So like, small pharma are doing discovery, and big pharma are buying them and like doing sales, marketing, distribution for them, right? And so that's irrespective of either one happens to be using AI or not, right? Like today, even just medicinal chemists, not using deep neural networks running in the cloud, but using deep neural networks running on meat, right, in their skulls, like that's sufficient for small pharma to outcompete, in some cases, big pharma. I think that sort of that universe of molecules is so vast, and there's so many decisions in multi-parameter optimization that you have to like get a ton of stuff right. And there's a bunch of choices you have to make where there isn't necessarily data, right? You just have to use judgment. So these mouse models, right? Like there will be a point when you're discovering drugs where the mice tox was bad, the rat tox was good. Which one's better for human? Right? Like that's going to be like in the absence of massive amounts of data, you're going to have to make a judgment call, right? And the big pharma will make a different one, and so then you'll be valuable to somebody. Okay. The other thing about like big organizations, and this is big organization if you're at Merck and Bristol Myers, it's a big organization if you're at IBM and Google and Facebook, is actually like your startup, the small team, is competing against some other small team. And you're not competing against the whole organization, right? Because like there is a small team which is working on the target you're working on, right? Or if you are doing, if you're Waze and you're doing like driving instructions, right? You're competing against like the Google Maps team, right? Which is like 4 to 20 people. It's not 20,000 people, right? And so like if you're going after protein aurora kinase C, you're going after like the team which is going after aurora kinase C, right? And that's your competition. And those people have to argue against, I used to work for IBM, and so like every August my manager would go into a cage match against all the other managers and argue why they should get budget and they should get headcount, right? Like, but it wasn't that all the organizations were kind of thinking the same thing we are. And so like that's, I think, the major advantage that the small pharma has is remember, you're actually not, it doesn't matter that they're big.